Dear students, in today's class, we are going to discuss the development of male gametophyte from the pollen grain. It's the process of development of male gametophyte, which is called gametogenesis, wherein a haploid microspore or pollen grain, which is produced from a microspore mother cell, is going to produce a pollen tube, and inside pollen tube there is a development of male gamete. Pollen grain is considered as the first phase of gametophyte, and the microspore mother cell is the last cell of sporophyte. As soon as microspores are released from the tetrad, they are referred to as pollen grains. Pollen grains are contained in microsporangia or pollen chamber. They have the size approximately 0.0252 to 0.125 mm. A freshly formed pollen grain is rich in cytoplasm with prominent nucleus, which is situated at the center. It comprises of two layers, outer exine and inner intine. The exine is thick, tough, cutinized layer which is often provided with spinous outgrowths, or sometimes it is smooth. The exine is composed of a complex substance called as sporopollenin. The sporopollenin has a waxy coating which makes pollen more or less waterproof. Sporopollenin is resistant to physical and biological decomposition, and due to this property, the pollen grains are often preserved for long periods in fossil deposits. The pollen wall also protects pollen during its journey from anther to the stigma. At one or more places, this exine is lacking. These areas are known as germ pores or germ slits. If these pores are round in appearance, then we call them as germ pores. If they seem elongated, and then it is termed as germinal furrow. Pollen grains are generally tricolpate with 3 germ pores, specifically in dicots, and in monocots they are called monocolpate with single germinal furrow. Germ pore facilitates the emergence of pollen tube through it at the time of male gametophytic development. The next layer present in pollen grain is called as intine, which is present inside. It is a thin, smooth, delicate pectocellulosic layer lying internal to the exine. Pollen kit is an oily layer forming thick viscous coating over the pollen grain of many insect pollinated plants. Pollen kit is believed that it may be contributing in the following ways. It acts as an insect attractant. It provides protection to the pollen against the damaging effects of ultraviolet radiations. It acts as an adherent to the insect body because of its stickiness. Being hydrophobic, it might even be associated with dispersal of pollen grains. It also functions as a pollen-borne substance involved in sporophytic incompatibility. Now let's discuss the structure of mature pollen grain. In over 60% of flowering plants, mostly dicots, pollen grains are set from microsporangium at two-celled stage, which are tube cell plus generative cell for pollination. Generative cell divides after pollination, and in the remaining species, pollen grains are set at three-cell stage, that is tube cell plus two male gametes. Generative cell divides before pollination. As we can see here in this picture, there is larger vacuole present here. There is the presence of generative cell, a tube nucleus with three germ pores. Once the pollen grain is released from the microsporangium, through the different pollinating mechanisms, pollen grain reaches the stigma part of the flower. Once it gets the compatible stigma, it starts sucking the nutrients from the stigma, and then the tube or vegetative cell elongates and forms a long slender pollen tube by coming out of the pollen grain through one of the germ pores. Generative cell immediately divides mitotically to form two non-motile sperms called male gametes. As we can see, this generative cell is present in the tube, which is leading or guiding the production of this pollen tube, followed by two male gametes. Thank you.